Okay, so from the last lecture, we had talked about the types of fast control to achieve homeostasis. And so we gave you three big examples with a minor deviation. And the first example here is an example of inhibition called product inhibition, in which what you're looking at here is what you're producing from that enzymatic reaction ultimately ends up stopping the further reaction of more substrates or nutrients. In other words, whatever you're making ends up prohibiting or stopping the actual enzyme from working any further. This idea of what we call a clogging the active site. In other words, when the reaction takes place, the actual product doesn't leave. Rather than giving the spot for something else to occupy, it is in the way, it doesn't let anything else in, and so therefore the concentration of the product will drop simply because there is no more substrate that can access the actual active site. And so eventually, as the products drop in concentration, then those products can leave, and then opening up the active site one more time and allowing it to continue its production. So that's one version of inhibition. The second inhibition is referred to as a competitive inhibition, in which now rather than the product clogging the site, now there's something else competing for that same site. In other words, two different things can fit in the same active site or in the same pocket, and each one of them having a different affinity, a different interest, if you will. And so what will happen is, depending on which one gets in there first, will prevent the other one from getting in. And then the last one that we're going to talk about with its subset is called an allosteric inhibitor. So here what we're talking about is when an inhibitor binds at a different location than the active site. So it no longer goes for that pocket. It no longer goes for that same place where the substrate fits in. Rather, what happens is it'll bind at a different location, hence the term allos. And what happens is it changes the shape of the enzyme. And remember that all enzymes work by their three-dimensional structure. So if something comes in and changes its shape, it stops being able to do what it does. Now, interestingly enough, there is such things as allosteric activation, in which changing the shape of the protein makes it work better. And so this is the idea behind cofactors and coenzymes, for example. Now, the last subset of allosteric inhibition is called feedback inhibition. And so here what we're looking at is usually in a series or sequences of events. And so when multiple products and multiple substrates are being produced, I'll show you an animation below, what happens is the last one in the series, typically at the end of the production line or of the assembly line, if you will, can go back to the beginning of the series itself and inhibit allosterically the very first set of reactions. The example that I can give you, and then I'll give you a visualization behind it, is when glucose undergoes those 6 types of steps that we had mentioned before, we'll cover those in a moment, is that the very, very last step in that production can actually go back to the very beginning and stop everything from working. So here, let me give you an example. So let's pretend that that little blue dot is an enzyme. And within that blue dot, we have our active site, the pocket where everything works. Now, traditionally, a substrate will go in, a nutrient will go in, and then when that nutrient goes in, it gets converted, it gets reacted, something happens to it, and we get a product. Now, that product will go on to another step, another enzyme, and that product becomes a new substrate and gets converted into something else. So a new product, and so on and so forth. We can do this multiple times, kind of like in glycolysis, except you'd have 6 steps. But then what happens is that very final product that you make now can go back all the way to the beginning of the steps and ultimately inhibit the first step, preventing it from working any further. And so in this case, this is called feedback inhibition, simply because the last step or some of the final steps in this series of reactions feeds back into the loop, preventing the first series from going any further and therefore inhibiting the whole process.
So does it technically contain an allosteric inhibitor then?
Correct. So
What's a good way to tell the difference between those two inhibitors? Like if you give us, like if we have to differentiate between the two.
Technically, you don't have to differentiate because ideally, an allosteric inhibition is included, the other way around, the feedback inhibition is included under allostery. So it is both. This is just a different effect in which when you have a chain of events, the final one comes and inhibits allosterically, whereas the previous example that I gave you, it's the same one step that gets inhibited allosterically. In other words, the only difference between the first one and the second one that I just showed you is that this one has a series of steps involved. That's the only difference. Does that make sense, Chelsea?
Yes.
Sense a little hesitation in there.
No, I just forgot I muted myself and I didn't want to make you think that I was ignoring you.
Thank you, I appreciate that. All right, so obviously we showed you a bunch of slides put together. So kind of let's try and show you what more or less I mean visually. It usually makes a little bit more sense this way. So in our first case here, we have an enzyme that is drawn out in roughly orange or yellow. And then I'm showing you what a substrate would look like in blue. Now again, that pocket where it fits the active site, the substrate itself fits in, creates this so-called enzyme substrate complex. And what looks like a hydrolysis reaction, taking a polymer and breaking it out into two smaller monomers, you get two little products in green and light violet or so. And remember that your enzyme always gets recycled, therefore it can redo this again and again and again, okay? So let's set up our first situation here in which we have what we call a competitive inhibition. And remember that competition means something else, something different gets into the pocket and inhibits it. In other words, something else gets in the way. So in this case, this gray inhibitor blocks the active site, preventing the original substrate, the blue one, from getting in. It's competing from the same spot. Everybody okay with this? All right, so let's take a look at the second one. In this case, allosteric. In an allosteric case, remember, it's a completely different location. So in this location, it changes the conformation of the enzyme itself. So remember, it's all about the 3D shape. And if it changes the shape, now our original substrate can't fit in, and therefore, again, we cannot make any more product. And then the last one that we were dealing with that kind of includes this system is our product inhibition combined with feedback. So here what we have is the product. So for those of you who recall, the little green one on the top can come back and either feed back into a series of reactions or just interact with the enzyme itself if it's only just one step, and then change its shape, also preventing the enzyme from working on that substrate, that nutrient that originally was the blue one, and preventing more product from being made. Now, typically speaking, Louis, you just asked that question. These other types of inhibition, not the competitive one, are usually referred to as non-competitive inhibition. It's still inhibition, you're just not competing for the same site. So, for the sake of just kind of racking your brain just a little bit, I wanted to provide you a few different examples of what this could look like, and these are meant to be a little bit more challenging than usual. So in case one, you can see the clear-cut idea between the substrate and the inhibitor competing for the same spot, a classic case of competitive inhibition. But what happens if you look at number 2, for example, here, in which you have a oddly shaped inhibitor and the substrate that can't get in? What type of inhibition would we be talking about now?
Competitive.
If it's a competitive type of inhibition, it would go for the same spot. And remember where the substrate is going. So since it's going to a different different active site, just like Jacob just said, this is an allosteric type of inhibition. It's going for a different location, preventing the first one from fitting in. But let's make it a little more complex and look at example 3.
Is that also allosteric?
Competitive.
I like how we're getting both answers because technically the answer is both. Because yes, you definitely have allosteric. That's absolutely correct. It's different sites, but you'll also notice that it's competing for one site in common. So it's partially competitive and partially allosteric. Now let's look at 4. Any thoughts?
Allosteric.
Definitely allosteric. So here, notice that they're not competing from the same space, even though one is kind of getting in the way of the other, they're not attacking the same active site. So if one gets in the way of the other, this is an allosteric type of inhibition. Now 5 comes in a little 3-part system, so you can take a look at it. What type of inhibition are we looking at here? Sherry, in this case, you're absolutely right here too. It's just a classic allosteric inhibitor. One changes the conformation of the other, preventing the other one from fitting in. Pretty standard, okay? Okay, so why is it that we look at these type of things? Why is it that we're paying attention to these inhibitions, these slow or fast methods? Because this is the classic case on how medicine works. Most of the time, our enzymes or other proteins in our body react by interacting with other substrates or reactants. And so what happens is medicine, therapeutics, designs substances that are designed to either act as inhibitors, either competitively or even allosterically most of the time. In other words, we create substances, substrates or reactants or nutrients that resemble the original substrate. So what do we do in the case, for example, in terms of antibacterial drugs or any type of antimicrobial? We can create drugs that mimic or look like the food for that particular organism. And so what happens is that organism takes up that food thinking that it's its own food, clogs up its active site one way or another, competitive, product, and so on and so forth. And then it dies simply because it's not consuming what it needs. So one of the biggest issues here is that we create drugs as a way to kind of kill these guys off. The problem with this is that evolution kicks in. And when this happens is that a lot of the organisms will die, but there's always that 1 or 2 that have a different way of surviving. And so this issue with this exploitation that we're dealing right now with antibacterials, so this whole set of people just buying massive amounts of antibacterials for no particular reason, is the issue that antibacterials also developed evolution. Evolution, remember, is just change over time. And so what happens here is that some of these organisms, some bacteria that do not have that necessity for that particular enzyme can just continue to survive while you just killed 99.9% of all its competitors. In other words, you removed all its competition. So now it has a free-for-all. And it can do this in several different ways. I'm presenting only 4 different examples here, in which sometimes the organism can just stop the production of whatever targets that enzyme. And so if there's no enzyme to target with your drug, it doesn't need it and it can survive. So your antimicrobial will not work. Sometimes it can actually take that drug and for some reason that enzyme can actually modify that particular therapy and now it does nothing to it. Other versions of them sometimes is having versions of the wall or membrane to prevent the input of the drug. So therefore that organism doesn't really die because it never takes it in. And probably the worst case scenario that we have is that it learns to more or less inactivate the drug itself. So it takes it, it goes to its target enzyme, but it somehow changes the drug and sometimes when it changes that drug, it can actually change it into something that's toxic for you. So not only do you give it the right drug, it changes it, it deactivates it. And then when it deactivates it, it now turns it into something toxic and kills you. Okay, so now that we've kind of had a live example of how these inhibitors are important to us, let's actually take a look at the actual pathways that we're interested in. And so this should be a relative review for all of you who have taken your good old gem biology and definitely your AMP. So, however, what we're going to do here is kind of do a summary of them, not really deal with them in detail, but the premise here is to just kind of get the gist, the important parts of what happens and why. That's about it. Why? Simply because here's medical micro, this is microbiology. We're not really covering all of the organisms out there. We're simply out here to understand how do things like bacteria and even viruses, not that they usually metabolize like this, use nutrients and sources to obtain their own energy. So, as we had mentioned to you at the beginning of this topic, the definition of metabolism by itself is referred to as everything that is going on as a chemical reaction inside a living thing. And so that's usually broken down into two classic pathways known as the anabolic and catabolic pathways, in which the anabolic ones, the term means to build. So this means these are the reactions that take nutrients or monomers, if you will, and create polymers. So this is your classic dehydration synthesis of creating substances that are larger. So when we looked at topic B, and we looked at chemistry, we started taking amino acids and turning them into proteins, or we took nucleotides and built them into nucleic acids. That is all an anabolic process. And every anabolic process that we know, including some of the ones as we built our macromolecules, always involve the use of energy. Building things, like anything else in the world, takes energy. Now, the opposite to it, its converse type of reactions, the catabolic ones, are all about breaking things down. And so this, in this case, uses hydrolysis as its main reaction. And its idea is to take larger things, like polymers, and bring them down to smaller pieces. And what's unique about this is that anytime we do that, there's a side effect of production of heat. So things get warmer when this happens. So organisms that perform catabolic reactions in high amounts usually generate some sort of heat. But the other side effect, the one that we really are interested in, is the energy production. When we break things down through hydrolysis, we can actually release small amounts of energy to use, including that heat as well, of course. So why is it that really this is the whole theme of this class, is that we're interested in not only the chemical energy that we can extract from these metabolic reactions, specifically the catabolic ones, but also for the fact that those types of energies that we harvest can be used to move things around, or to either build things or transform things within the body. And so for those of you who recall learning this from previous classes, the way we talk about energy is not really as energy itself, because that's more of an abstract type of concept, if you will. We talk about what carries the energy. And so, as we talked about in the quiz 2, there's two main types of energy carriers out there. And probably the most obvious of them all is what everybody knows as ATP, or adenosine triphosphate, which is a storage of energy, if you will, as a result of adding one more phosphate to the adenosine. And then we have the second version of one, which is called nicotinamide adenine dinucleotide, or NAD, which happens to have a positive charge. And you'll notice that I have the 2 highlighted there, and I have a little asterisk next to it. It's because there is a slightly more varied version of NAD called FAD, or flavin-adenine dinucleotide, which virtually acts in the same way and does very similar things to that for our courses, with the exception that it's a little bit weaker. So we kind of group those 2 things together. NAD and FAD are put together, and then adenine triphosphate is the second one that we have there. Now, how do we produce these? And what's the purpose behind these, aside from carrying energy? Well, the most basic concept that we get to learn overall in this class is the subject of one of those 6 chemical reactions we talked about in topic B, which is phosphorylation. Now, the idea here is a very simple concept: to add a phosphate group, one of those functional groups that we covered in topic B as well, is that third and final phosphate, when added to a particular substance, for whatever reason, is what we call a high energy bond. In other words, that one being contained in there, when removed again, releases a lot of energy. And so the example we're going to use here is ATP, in which when we add that final and third phosphate to adenosine diphosphate, creating this so-called adenosine triphosphate, we create a storage of energy in that final and third phosphate. And pretty much what you should be thinking about it is as if you had a battery. In this day and age, we transport most of our energy by the use of batteries. So whether it's your phone, computer, or anything that is portable, it usually contains some sort of storage or storable source of energy. Those are what we call batteries. So energy carriers, in essence, are that. It is a form of a tiny little packet of energy that we can transport from point A to point B, so we can use it at a later time. In other words, we can create it, store it, and use it whenever we need it or wherever we need it. And so what we're going to learn in this class is how to generate that battery. How do we create those energy carriers? And there's three classic types that we learn in most gen biology classes. The first one is called substrate level phosphorylation, or SLP. The second one is known as electron transport level phosphorylation, or ETLP. And then the third version, which we'll look at much later on today, is photophosphorylation, and that one doesn't usually get abbreviated. So let's look at the first one. Since we've been talking about lots and lots of reactions, the term substrate should kind of be the dead giveaway here, is that we're looking at something that contains a high amount of energy, usually those phosphates, and that it's transferred to the substrate, in this case, ADP, or adenosine diphosphate. So in essence, we're taking some sort of substance that is energy rich, so we'll call that a donor, and that energy rich donor will transfer it, to adenosine diphosphate, creating that third and final high energy bond of phosphates, creating our ultimate energy carrier. That's the process of substrate level phosphorylation. Now, for those of you who are not paying attention to my amazing drawing of our donor, you should get an idea of what shape does that look like. Most high energy donors happen to be things that carry massive amounts of sugar in them. So carbohydrates happen to be our best donors for us. And as we will learn in a few more slides, we'll look at the very first set of metabolic pathways, which is glycolysis, which literally uses glucose as its first step to create energy. Now that's version 1. Version 2 is known as the electron transport level phosphorylation. It does have a second name called oxidative phosphorylation, if you will, depending on which book you read. And the premise here is two key steps put together. The oxidative level indicates the transfer of electrons. And so what's happening here is you remove some electrons from something that is also high energy, and that electron gets transported through these carriers, usually called cytochromes, and this exchange of electrons taken back and forth is the process of oxidation. The loss of electrons is oxidation. Good old chemistry reminder. Now, the problem with those electrons is that because they do possess high energy, they usually need to end up somewhere. And that somewhere goes back to our nutrient class, our nutrition class, when we talked about oxygen being a terminal electron acceptor. Typically, oxygen that loves electrons is the final guy that captures those electrons. Now, the second portion of ETLP is the phosphorylation part, because what will happen is then after this exchange of electrons back and forth, that oxidation part, we harness enough energy to actually be transferred to the conversion of ATP, to the building of ATP in the process of phosphorylation. So when we get to that step, we'll look at all the details behind that one. Now, the last and third version of generation of energy is photophosphorylation. Now, here the detail is actually quite simple. It is very, very similar to the previous step. So it's very similar to the electron transport level phosphorylation. The only difference here is that we don't have a substance that contains the electrons in the high energy. Instead, we utilize sunlight. And sunlight usually is the one that strips off the electrons by hitting them with photons, the sunlight and the radiation that's coming from the sun. So that also results in the formation of ATP. So ETLP and photophosphorylation are relatively similar. It just depends on where the electrons are coming from and how they're made. And again, we'll take a look at them in a few moments. All right. So the rest of this topic is solely based on covering the actual pathways. And again, these are summaries of the pathways themselves. We're not going to go into any details. I'll show you what they look like, and I'll tell you exactly what I want. So I strongly encourage you to kind of map these out on your own so you can have an idea of what these mean. So I'll show you some examples too. And we can start with this with the catabolic pathways. We're going to look at glycolysis and its 3 variations in a moment. We'll look at fermentation, and then we'll ultimately look at both types of respiration, aerobic and anaerobic respiration. Okay. The idea here is the reason why we look at this is because, again, we're looking at it from the perspective of microbiology, not just humans or animals all over the place. So let's look at the very first step in its generalized format, glycolysis. So you'll notice that in my tables, I've created kind of a summary of what the whole thing is. I can tell you what the purpose behind it is, which glycolysis is as simple as breaking down glucose into simpler substances to extract some energy from it. The location is important, so the where, and so this takes place free floating within the cell, so inside the cytoplasm. And then what goes in and out of the actual set of reactions. Okay, so what we have here is the conversion of 1 unit of glucose. It gets broken down, it kind of gets split in half into 2 units of pyruvate or pyruvic acid. And then you invest a couple of pieces of ATP, 2 of them in particular, 2 energy carriers, for the ultimate production of 4 units of ATP. So there's an investment of 2, an extraction of 4. However, in order to produce these 4 units of ATP, there is a small loss of the other energy carrier, the NAD, which ends up being used or used up or reduced is the term into a form called NADH. Okay, so to kind of summarize this to make it a little bit easier on you, I'm taking that same table, I've kind of shrunk it, still shows the same thing. And for as a reminder, or more or less a quiz for everybody, does anybody remember where we get our majority of our glucose from? Anybody?
Carbohydrates.
Most carbohydrates. And where do these come from in our diet? That should be the hint. No, nobody. Virtually everything we consume, as long as it has sugars in them, is full of carbohydrates, and in particular glucose. So what we're going to do in this set of summaries is that we're going to extract glucose from it, which happens to be a carbohydrate that is composed of 6 carbons. And ultimately what's going to happen is you're going to break this down, you're going to split this down in 6 reactions or so into 2 half pieces. These half pieces are referred to as pyruvic acid or pyruvate. So you've literally taken that 6 carbon sugar and split it off into 2 tiny little 3 carbon sugars. And while you're doing these 6 steps, you'll extract a little bit of extra energy from it. Now, if you look at it from your textbook perspective, you can see all of those reactions and what actually happens, the conversions, the investment of ATP, the extraction of the extra ATP, and so on and so forth. It's a little bit easier to just kind of highlight it in terms of really what it means. You're taking a 6 carbon sugar, breaking it down into 2 pieces of pyruvate. This will generate energy, use up or reduce the NAD in 3 classic stages. The first portion is known, the first half, if you will, is known as the energy investment stage, in which you have to pay 2 ATPs to make this. The third stage, the second stage here is the breakdown of glucose into 2 pyruvates. And then the third stage is the energy conservation stage, in which you're now extracting an additional amount of energy from 2 ATP into 4 ATP. It's easier to visualize this way. The beginning, you add some ATP to make this happen. This is that substrate level phosphorylation portion that we were talking about. We're forcing some phosphates in there, okay? We split it off, we hydrolyze it, we lyse it, we break it into half. We get 2 3-carbon sugars, and then ultimately we extract some extra amount of energy. Now, this process, as basic as it gets, has 2 major challenges. The first one is that we are using up some of our energy carriers, those NADs. So that's not good. But the second, probably the most important issue here is that those pyruvates, those pyruvic acids that we just formed after splitting off glucose by glycolysis, is that it's toxic. It's actually fairly toxic. So we need to find a way to get rid of it. So let's go back just for the quick summary. Let me kind of show you the slides. Back to where we started here. We're going to take a 6-carbon sugar, break it in half through several reactions into 2 pyruvates, use up some energy carriers to make more. So we used up some NADs and some ATPs to make 4 ATPs. But the problem with this is that those pyruvates happen to be toxic. So we need to figure out a way to get rid of them fast. So from this, what do we do with that particular pyruvate? So after glycolysis, we need to get rid of it. And so there's 2 versions that we're going to teach you on how to get rid of it classically. The first step is called fermentation, or the second version of this is called respiration. So we're going to look at both in order in this case. So kind of summarize, glucose gets broken down into pyruvates that happen to be toxic, and now we need to get rid of it. So we go back to our original format, the table that I showed you before. The purpose behind this set of steps of fermentation is to get rid of that pyruvate. Now, one of the conditions behind this is that this set of steps, these set of catabolic reactions, happen on what we call anaerobically, meaning these only take place in the absence of oxygen. Okay, and this still occurs kind of freely floating in most cells, regardless of whether prokaryotes or eukaryotes. So now, what is the main investment? Well, here the main reactant or substrate is pyruvate itself. And what's really unique about this step is that those NADHs that we produced in glycolysis will get recycled back to their original state. In other words, you'll convert your NADH back into NAD, turning it back into a useful form of an energy carrier. Now, the pyruvate, on the other hand, gets broken down, gets catabolized by removing one of the carbons from it. And this carbon gets then released as carbon dioxide. So let's kind of summarize this a little bit, visual kind of take. Okay, we've shrunken the table. And now we're going to start with just one. Remember, we created 2 in glycolysis, but we're going to only look at one of these 3 carbon sugars. And what will happen is we'll extract one of those carbons, we'll break it down, and that 1 carbon allows us to leave only 2 left and turn in what we call a less toxic compound. So something that is not as bad as pyruvate. And there's 2 options. The first option could be ethanol, and the second option can be lactate. So now, what's interesting about these are separate methods. Ethyl alcohol or lactic acid are the classic 2 types of results. So let's take a kind of look at data why this is important. In some of these versions is that when we break down this fermentative step is that we recycle our energy carrier. So we got back our NAD. So that's a good thing. The carbon dioxide that we produce here happens to be readily diffusible, so it goes away, leaves all our cells, so it's non-toxic either. And thankfully, the compounds that we produce are not as bad as pyruvate. However, they still have some toxicity associated with them. Now, what we're going to look at here is kind of the summary of those reactions, either the production of ethanol or the production of lactate after glycolysis. So in the top portion of each of these reactions, you can see glycolysis happening, the creation of 2 pyruvates. And in the bottom portions of these reactions, you can see the production of either lactic acid or ethyl alcohol. Now, why do we look at these specifically in microbiology? Well, for 2 reasons. One is because we look at organisms that are anaerobic, meaning that they do this in the absence of oxygen, meaning that oxygen usually kind of is toxic for this and prevents this from happening. Now, the 2 versions that you know are actually quite important industrially speaking. Why? Because in one version of this, the production of ethanol is a very common effect that we use in most industries as a production for things like alcohols, so our classic beers and wines and things like that. But also organisms like yeasts, for example, can also produce gas through that fermentation, And this is what allows our breads to rise, for example. So your classic sourdough bread, that kind of thing comes from there too. Now, it's an interesting fact because technically humans can ferment too for a very, very small amount of time. And this is a very small amount of time. Now, the problem is that for us, when we stop breathing, especially when we're working out or something like that, we don't have oxygen, and that oxygen lack creates an anaerobic environment. For us, when we undergo this, we actually ferment lactic acid, or in the case, lactase. So what's the issue here is that this is a signal that causes our muscles to lock up. Contrary to popular belief that people think that this is what causes the burn. For example, when you're working out, the burn that most people feel is as a result of micro tears in the muscles. Lactic acid is actually part of feedback inhibition. That lactic acid will tell our brain and our muscles technically to stop working out before you actually damage them, specifically because you're not breathing. So that's kind of an important thing in here. So there's an industrial application that we're interested in here that allows us for the production of certain products because of fermentation of pyruvic acid or pyruvate, or in this case, as signals inside of humans. Thankfully, I assume that most of you would be probably happy that you do not ferment ethanol, because then the entire world population would cease to exist. I'll let you think about that in a moment. So one of the options here is that glycolysis, producing pyruvic acid, produces something that is toxic. And so one of the options, in the absence of oxygen, an anaerobic condition, we can ferment it. However, thanks for the fact that we at least have roughly 20% oxygen present in our air, lots of animals and plants and things like that can actually utilize this and undergo a separate different step called aerobic respiration. Aerobic respiration gets broken down into two key steps. These are known as the Krebs cycle and the electron transport chain. So we're going to look at both of these kind of side by side. Now, the first one, the Krebs cycle, is a series of 8 reactions or 8 main steps with some deviations in there that are all designed to just break down pyruvic acid. Now, what's interesting about this is the requirement that this has to happen inside the mitochondria of eukaryotes, or if your cells do not have mitochondria, this typically happens in the plasma membrane. And so the key feature here is that we're going to take pyruvate, one of those, remember you produce 2, and break it down to its ultimate completion. The problem with this is that it costs a lot of energy. Remember, this is toxic for us. So we're going to use 4 molecules of NAD, 4 of those energy carriers, and usually 1 molecule of FAD involved in this, for a grand total of 5 of these energy carriers just to do this. Then what you'll see in the products is that ultimately pyruvate completely disappears. And even on the side, we managed to make 1 small unit of ATP, 1 extra little battery for the use. Now, what this looks like, for those of you who are not familiar with, is a giant cycle, hence the term Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle. They all mean the same thing. It's a complex set of reactions, a cyclical set of reactions, in which each step breaks down a little bit of pyruvate until you remove all the carbons. So let's give you kind of a better idea of what this looks like. What it's really kind of turns into is this series of steps that all kind of communicate the breakdown of pyruvate into virtually all carbon dioxide. During the very first investment stage, you actually end up stripping off the first carbon and turning it into a true carbon sugar. And so those carbon dioxides go away. Now, for the first 3 sets of reactions in there, you'll actually strip off another carbon dioxide, leaving it down to just 1 carbon sugar. And then you'll do this again for a couple more reactions, and now converting that final remaining carbon sugar, or 1 carbon sugar, and turn that into CO2 as well. And then the last set of reactions will use their components and bring it back to the beginning of the actual cycle. Now, while this is happening, you end up using 4 of those NADs, 1 at the beginning, 3 in the middle, and 1 of those FADs to make this happen. And towards the end, you end up producing a molecule of GTP, which eventually is kind of roughly equivalent to a unit of ATP, or an energy carrier. So this is kind of like a game of telephone. Each time somebody's passing down some information, but every single time somebody passes it on, something is lost. Here, what you're losing is a carbon, and that carbon turns into carbon dioxide. Now, reminding you that this started off as 2 pyruvates at the end of glycolysis. So technically, when you look at this, you kind of double the amounts. So we take our 2 3-carbon pyruvates, run this cycle, and ultimately, we'll use up a grand total of 8 NADHs and 2 FADHs produced from this recycling process. And at the same time, we will extract 2 units of ATP. Remember, this is being doubled. Now, the key feature here is what I want you to understand is that every single one of those carbons got turned into CO2. And since you started with 3 carbon sugars, you created 3 CO2s. But since you ran this twice, remember you started with 2 pyruvates, you generate a grand total of 6 carbon dioxides. Now, what does this mean? The overall steps behind the Krebs cycle, the great benefit behind this, is that every single one of those carbons from glucose that ultimately turn into pyruvates now got turned into CO2. In other words, you turn all the glucose that you ate into carbon dioxide. And on the same side, you actually create a little bit of energy on the side. The problem with this all is that you did use up a bunch of those NADs and FADs. And it's kind of important at this point in time to actually track how many of those have we generated so far, not only from this step, but also from glycolysis, in which we actually generated a few of them. Okay, so what is the big deal behind the Krebs cycle? Just like fermentation, you're getting rid of pyruvate. So that's a great thing. And just like in fermentation, sometimes you can produce a little bit of extra energy. But the great thing about the Krebs cycle, though, is that we can get rid of all those toxic pyruvates into CO2. So in the bigger picture here is I want you to understand that as long as you are breathing right now, which I assume you are, is that you will choose to invest your pyruvates that you're consuming from all that food that you're eating at home, and it'll turn into pyruvate. And then all you do, as long as you're moving and using your lungs, you'll turn it into CO2 and exhale it. You'll breathe it out. So every single time you're consuming those carbohydrates, those sugars, those glucose molecules, they all turn into that good old CO2 that you're exhaling right now. Now, I told you the respiration, aerobic respiration is a 2-parter. The second part is the one that usually ends up being a little bit more complex for people. We'll kind of simplify it for you. It's called the electron transport chain, or also known as the ETC. Now here what we have is the whole process of just recycling all of those used up NADHs and FADHs. This also takes place in the mitochondria or in the plasma membrane of cells. But the key condition here is that it has to have oxygen. Remember, this is an aerobic condition. So no oxygen, this all falls apart. In other words, you don't breathe oxygen, you die. That's the same premise, okay? So the only key reactant that you need to know from here are those reduced or used up energy carriers. All those NADHs and FADHs we've been producing in the past set of catabolic reactions. And all that we do is recycle them back to their original state. So this is definitely the benefit of the electron transport chain. Now, how do we look at this? It's actually kind of an interesting concept that we looked at a little bit earlier, is that those NADHs that we used up earlier are full of electrons, which means that technically they're energy rich. And so what we're gonna do is typically inside mitochondria or in plasma membranes is we're gonna transport that NADH in, use it up in there, extract those electrons as a way to create energy. So what's the story first, and then I'll show it to you visually, kind of summarized. Here what we're gonna do is we're gonna take all those electrons that are present inside those NADHs and FADHs and strip them off it. And really what we're gonna do is take those electrons and pass them on to oxygen. Remember, the key ingredient here is oxygen. Oxygen being a terminal electron acceptor, or this highly electronegative gas that loves electrons. It will steal those electrons from NADH ultimately. Now, what happens while this is occurring, those transfers of electrons, a little bit of energy is being given off. So this turns into this little chain of events that creates lots and lots of hydrogen ions or protons kind of being moved between the membranes of the mitochondria, creating what we call a gradient. So that gradient then ultimately creates a power source to create more ATP. So let's kind of summarize again. What typically happens is that while these electrons are being exchanged, hydrogen ions are moving across membranes. This creates the so-called gradient that I was talking about. Now, if you want to think about it this way, the easiest example I can give you is that gradient, meaning there's one side, there's more than the other side. Think about it as if it was like a water dam. On one side, you have the water building up with massive amounts of energy, ready to cross to the other side. And on the other side, there's nothing. The dam, being the one that splits off everything, like our membranes, contains that gradient. And then ultimately, as we allow some of that water to pass through, just like we would use in dams also, we can install things like turbines. And turbines, as the water passes through, powers those turbines and we create electricity. Well, the same premise happens here. Our turbines are proteins called ATP synthetases or ATPases, or ATP synthetases, I'm sorry. And so these little turbines, these synthetases, allow some of those protons to go through, powering that protein to create ATP. So let's kind of look at it this visually. It makes a little bit sense kind of in pretty pictures. So we start at the bottom and we looked at our NADH that we used up, the one that we really need to recycle. And so what happens is when we recycle it, we strip off the electrons, converting it back to an NADH. And that guy is recycled and ready to go. However, those electrons need to be transferred somewhere. And so that electron will pass through a series of proteins called cytochromes. And as it transports it, those cytochromes will start pumping out hydrogen ions or protons from one side to the other of the membrane. And then at the end, that little electron will land on oxygen, charging it, and then that oxygen trapping that electron can now combine with some of those hydrogens and create water. Now, all while this is happening though, you keep on pumping hydrogen ions on the other side, creating a gradient. A lot of hydrogen ions are present on one side and a small amount on the other side. In other words, you created that dam in which on one side you have water and the other side you have very little. And so that gradient creates this ability to transport from one side to the other. Remember diffusion, that concept that we learned during the cell lecture, is things want to go from high to low. So since they can't cross the membrane because they're electric, they're charged, something has to let it through. And what lets it through is a protein called ATP synthase or synthetase. And so that's our turbine. So as we let the water flow through the dam, through these turbines, we actually create energy. Here, instead, we're creating molecules of ATP. So as that gradient gets created, some of those hydrogens pass through, 2 to 3 of them at a time, and we use that to power the adenosine diphosphate and the phosphate units to create an ATP unit. In other words, we're creating energy just like if a dam would allow water to pass through, except that here we're using hydrogen ions to do the same thing. So what are we looking at here? The idea here is that the water that we're creating with the oxygen and hydrogen ions isn't toxic for us. We are recycling all of our NADHs and FADHs back to the original state. So that includes both of the energy carriers. And remember that as long as we have oxygen present and we create that gradient, we're actually making more ATP. As a matter of fact, each ATP that each NADH that we recycle ends up producing roughly about 3 ATPs. And each FADH, which is a little bit weaker, that is recycled, creates about 2 ATPs or so. So here it's kind of important to understand how much. And so as long as you're kind of keeping track of this, you can actually do a little bit of math, and we'll do it over here, just kind of keep track. Is remember that during glycolysis, I'll kind of summarize these in certain steps. During glycolysis, right, you took in 2 ATPs, and created 4. However, you did lose 2 NADHs there. Same thing during Krebs cycle. You ended up losing about 8 NADHs in total, and actually 2 FADHs. FADH2s actually. So if you do the math, we ended up losing 10 NADHs and 2 FADHs here. So when we make it to the electron transport chain, and if each one of them that gets recycled produces 3 ATP or 2 ATP, we actually have a really good turbine. This turbine that is producing ATPs, out of the 10 NADHs that we recycle, we generate about 30 ATP. And then out of the 2 FADHs that we were recycling, we generate about 4 more ATP, for a grand total of 34 ATP recycled from those NADHs and FADHs. That's not to include the extra 2 ATPs that you produced from glycolysis. So that's 2 more net. And then the 2 extra ATPs that you produce during the Krebs cycle as well. So just for the virtue of having oxygen present, we can actually recycle all of the stuff we used up earlier and create roughly about 38 to about 40 ATP per each 1 molecule of glucose. And this is really where this device becomes critical, as opposed to everything we've been learning so far, like fermentation, for example. This is the main pathway. Glycolysis, Krebs cycle, and electron transport chain generates a massive amount of energy with a small amount of heat loss in there to allow the growth and development of most organisms. So glycolysis combined with respiration is a very powerful tool, as long as, again, you have access to sugars, technically glucose, and you have access to oxygen. When oxygen is absent, most of this is not possible, and it becomes very energy inefficient. Now, kind of putting this all together, remember that our electron transport chain works by virtue of these carriers, these cytochromes, and pumping out this hydrogen that creates that gradient. That this takes place either in the mitochondria or the plasma membrane when you're talking about bacteria, okay? Now, our bacteria, though, because they are using their plasma membrane as a way to do this, there's some variations. So they're not as simple as using just mitochondria for us. And so different bacteria have variations of their cytochromes, for example, and they actually have different enzymes that work at different temperatures as well. So they have some variability. Nonetheless, virtually all of these use oxygen as a terminal electron acceptor. However, some organisms can utilize something else instead of oxygen. And for those of you who can visualize in your mind the periodic table, you can actually use other substances close to oxygen as also electronegative substances or elements as terminal electron acceptors. So if you had oxygen, for example, and you create H2O out of it as a final product, if you use sulfur instead, which is also closely related to oxygen, instead of producing H2O, you produce H2S. But this is hydrogen sulfide. So these are our sulfur-producing bacteria. So you can use nitrogen, you can use other versions of methane, and so on and so forth. So oxygen is not the only electron acceptor. That's the one we utilize. However, there's other versions and there's other Ts that you can use. This is where our variation begins. So let's start off with the first set of pathways. Glycolysis, as basic as we just learned it, has some variations. So the one we looked at is called the Embden-Meyerhof set of glycolysis, the one that you and I utilize all the time. However, there's 2 other versions that are out there. The first one is called the pentose phosphate pathway, and the second one is called the Entner-Duderoff pathways. Now, what these are still glycolysis, okay, but they just kind of vary a little bit of it. For example, the pentose phosphate pathway utilizes pentoses, so 5 carbon sugars instead. And so they can use sugars like ribulose, xylulose, ribose, that kind of thing. And so they still undergo glycolysis, but they produce a little bit less energy. Now, the other thing, what it does do, though, is that by doing so, some of those components get used to turn them into what we call precursor metabolites, meaning we use them for building materials instead. So their glycolysis is less efficient, but they do produce building materials. So there's a little bit of an exchange there. Now, the second pathway that we learn about here is called the Entner-Duderoff pathway. Same idea, same premise. It produces a little bit less energy, but it also produces a different energy carrier called NADPH. So that P is for an extra phosphate attached in there. And this particular pathway of glycolysis is present in different versions of gram-positives and gram-negatives, and again, produces precursor metabolites and a little bit of less energy. So variations in the actual production of energy. Now, to close this off, before we go into anabolism, most of the time people end up asking, what about using other sources of energy, which are typically your fats and proteins? And this is what we're doing in the lab. We're looking at how the organisms use things like proteins in decarboxylations. So one version of this is the breaking down of lipids, known as fat catabolism or lipid catabolism. And the idea here is you'll take some of those components and break them down and introduce them into glycolysis in the middle or into the Krebs cycle. Now what happens is you have these enzymes called lipases or lipid hydrolases that break down the fat into the glycerol unit, and that tiny little glycerol can be thrown into glycolysis. And then the fatty acids that are present in those lipids undergo a process called beta oxidation. Now, what this beta oxidation is those fatty acids get broken down into chains of twos. Remember, these are very long chains, and each one of those will produce a small amount of energy when it gets inserted into the Krebs cycle. Glycerol, on the other hand, the little pieces get thrown into glycolysis, and sometimes you generate a small amount of energy. Really, where the energy comes from is from that beta oxidation. Each 2 carbons that are present there generates a small amount of energy. The only problem here is that even though fat really produces a massive amount of energy when broken down, it also takes up a lot of energy to do so. So even though fats contain high amounts of energy, if you will, it's not our most optimal source to do so. Our evolution has dictated that carbohydrates are our best source of energy, most efficient way, and that fats, even though they can produce it, we ultimately like to store our energy as fat because it holds it well. So, contrary to popular belief, starving your body from carbohydrates actually makes the situation worse, especially during diets. And then finally, the other option behind fancy little diets that don't work are the use of protein. Diets that are heavy in proteins can utilize excess protein for small amounts of energy. And the reason behind this is that we can take some of the amino acids present in the our proteins, and they're broken down by enzymes called proteases, or protein hydrolases also, or protein deaminases or decarboxylases. And so they'll remove the amine terminus from the amino acid, and those will yield ammonia and ammonium as a nitrogen source. The rest of it will go into the Krebs cycle, and these will produce small amounts of energy. So we'll take our long chains of proteins, break them down into original amino acids, and those amino acids can then be thrown into the Krebs cycle for a tiny amount of energy. Proteins are not a very good source of energy at all. They just work in emergencies. However, one of the more important aspects behind this is the fact that when we're looking at the breakdown of proteins, is that they yield that ammonium. And what's the big deal behind this ammonium is that it can produce a compound called TMAO. Now, this stands for trimethylamine oxide. And trimethylamine oxide happens to be one of those signals that something is dying. So it is one of those instant smells that we know that something is rotting. And this is a classic fishy smell. So TMAO, for example, if you go to any fish market or butchery or something like that, that's probably the first scent that you receive. And so anytime you can smell that, you actually know that what you're buying is not fresh. Technically, if you're talking about things like fresh fish, if you will, should not smell fishy. That fishy smell that you all detect is from TMAO. So anytime you're buying something, you feel that smell, it's a bad thing. All right, let's close off the last few minutes here on the subject of anabolism, in which now we're seeking to build things, okay? And this is one of the more common misconceptions behind here, is that people think that the term photosynthesis is how they produce energy. The term photosynthesis should give you a dead giveaway in terms of terminology, in which photosynthesis means to synthesize something. So here's an anabolic pathway. This is the building of materials, not the breakdown of them. The breakdown of it is the portion of photophosphorylation. So our anabolic pathways here will look at our chloroplasts or any of their derivatives, their cousins, if you will, that'll use their membranes as a way to break down and produce energy and food for things like plants and algae and a couple of their variants. So again, the classic green color that everybody's familiar with comes as a result of something called chlorophyll, or its variations called bacterial chlorophyll, in which they're present within the third and second layers of the chloroplast. And so here, we're going to look at two steps: the catabolic portion of the production of energy, photophosphorylation, and the building of materials called photosynthesis. And so these are divided into what we call light reactions and dark reactions. Now, what these indicate is that the light reactions are all about producing energy, so that's phosphorylation. And then the dark reactions are all about producing food. So this is the photosynthesis part that everybody's familiar with. So we're going to look at both of them and their locations. Now, just because they're called light and dark doesn't really imply anything. It's just that the light reactions require sunlight, but the dark ones just means that they're light independent, meaning they can occur with or without sunlight. Doesn't mean it needs to be in the dark. So we'll look at our chloroplast again, and our dark reactions will occur between this third and second membrane, and then our light reactions will occur within the third membrane. And so our final summary here is fairly quick. We will look at portion one, which is the production of energy in the light reactions. So this is the use of sunlight in this case by using specific enzymes and specific compounds like chlorophyll. And they occurred inside the third membrane of chloroplasts, or their variations called thylakoids. And so the investments here are light, water, and a variation of our energy carriers called NADP, or NAD phosphate, if you will. So what happens here is that we'll use sunlight and water as a way to generate oxygen and energy. And then we use up the NADP feature. So we're going to look at our a similar sketch that we drew before, except that here we will take sunlight that will usually hit water, the key requirement here in most plants and algae and things like that. And that radiation, those photons, will strip off the water, hydrogen, and the electrons from the water itself. And so those electrons can then go the same pathway, just like we saw kind of in reverse in the electron transport chain. And those electrons ultimately will land on NADP, producing it, and at the same time, also spitting out hydrogens or protons between the membranes. Now this NADP will now carry those electrons and turn into NADPH, so it's used up. And then the rest, you already know the story. Because of that electron, we get this gradient on the outside that creates this dam that, when allowed to pass through, we can actually create energy again. So what are we seeing here? Is that here we're using, as long as we have water around, or something that is equivalent to water, we can have oxygenic pathways in which sunlight will split off things like water and create oxygen gas, or it can split off things that are not carrying oxygen. We call these anoxygenic pathways, things like archaea and some of our deeply branching bacteria, and release something like sulfur gas instead. So here, what's great about the light reactions is that you make lots of ATP, and they also make oxygen. Now, the only reason why that's a benefit is because of us. For those organisms that are aerobic, that's a good thing. It's not necessarily a good thing for the plants, it's just a side effect. The only problem for the plants, though, is that they're now using up their NADPs into NADPHs. So here's where the other portion comes into play. the dark reactions. And so the dark reactions occur within the actual stroma of the liquid present between the third and second membranes. The third membrane produces our little batteries, our light reactions. The third and second membrane space in between, the stroma, produces here food. So how does this work? It'll take those NADPHs that we had used in the previous set of reactions. It'll use up some energy, and it'll take up the CO2 that's freely floating in the environment. And what it will do is it'll take the NADPH and recycle it back to its original state. And it'll take all of those CO2s and convert them into glucose, what we know as starch or amylose. So what you end up having is a very similar situation like the Krebs cycle or tricarboxylic acid cycle, except that this one is called the Calvin cycle. And it's 12 steps instead. And so it's kind of the Krebs cycle in reverse. So let me kind of show you this to make it a little bit easier, in which now you invest CO2, undergo multiple reactions, recycle your NADPHs, and now you'll produce little molecules of glycerol, which ultimately will lead us to the production of glucose. So what are the results? Here, the ATP and ADPH that you did in the light reactions are now used to generate food for the plant. The products in this case, glucose, food for the plant, and ADPH are recycled, and now you end up having what we call a perfect organism. What we have here is an organism that, by possessing mitochondria and chloroplasts, for example, or some variation of chlorophyll, they can undergo light reactions to produce energy, and they can use dark reactions to produce their own food. In essence, we have plants or their variations that use CO2 to build sugars, and then they can take those sugars for food and build the rest of the plant or their variation themselves. And they can then use the water and sunlight to create all the energy they need for this relation between those two pathways. So those are all our photosynthetic sets of reactions that we need to do in terms of anabolism.